From your slides, nice to see you. Welcome over. I'm glad we have time to chat later and have dinner. So looking forward to it. Oh, oh yeah. Well, so you re-met him or something like that. Yeah. Good. Okay. So you is he? You met him? Oh, what? Oh, I just said hello. I didn't know if he was going to lunch, but it's before lunch. Okay. All right. Yes.
After, I'll come get you and bring you. That's why I thought we did. Oh, really? I'm seeing you out here.
Do you want to give me a signal when I should speak, or?
Sure, I'll go to the back
And do they want this or not?
it's probably on. It's
Yeah.
usually on.
I think it helps. You have feedback? Yeah. Well, you tell me. Hey, welcome everybody. Thanks for coming. It's my pleasure. Oh, maybe I'm supposed to use this. My pleasure to introduce our MIT neighbor, Yingxi Lin. She was an undergrad and did a master's also at Tsinghua University, and that was in engineering physics, and the master's was in something related. I can't quite remember now. She came to Harvard to do her PhD in the physics department. She did her thesis research at Longwood with Gerhard Wagner, doing protein structure using NMR. NMR is the only technique where you can figure out protein structure in solution. And she worked on some aspect of histone acetyltransferase in complex with something. Coenzyme A, maybe, yeah. And then she switched to neuroscience and did a postdoc with Mike Greenberg in the neurobiology or at the medical school. And there she worked on a protein that's been important to her since then. And at that time, she did a number of things, but as a postdoc, her research was on the role of this particular protein, HPAS4, in inhibitory synapse development. And she's been on the faculty at MIT for almost 10 years now, and she's been involved in a number of things, but really her work is on fundamental aspects of learning and memory, especially memory encoding, especially contextual memory, as we'll hear, and HPAS4 has continued to play an important role. So eager to learn about it. Thanks.
Thank you, Kenny. Let me move away. All right, it's working? Okay. Am I too loud?
No.
All right, good, perfect. All right. Well, even though MIT is not that far away, I feel I don't come over here often enough. It's my pleasure, and I'm very excited, especially today. I'm excited to share with you some of the new stuff from the lab, and I'm published. A lot of the stuff I'm showing you today is unpublished, and as you can see, it might not be very well rehearsed, but I like to get some feedback from you guys. From the title, you can see that my lab has been very interested in the heterogeneity and diversity within the memory engram. So memory engram is defined as enduring biophysical and biochemical changes elicited by learning. So in that sense, memory is the impression that our experience made in our brain. Right? So there are a lot of debate about what the engram is. So my goal here today is not to argue about what the memory engram is, but to show you like that what we have been focusing on to think about how memory is encoded in the brain, how the encoded memory affects the behavior output. So us and many people in the field of in modern neuroscience have been focusing on the neurons called active ensemble neurons as a strong candidate for memory engram. So these neurons are activated during learning, right? So this is everyone agrees upon. And the reason they are considered a strong candidate for memory engram is because that there are evidence suggests that there are some enduring biophysical, biochemical changes happening on these neurons. For example, there are a couple of recent studies clearly showing that there's Hebbian type of synaptic strengthening, experience-dependent plasticity happening on those active ensemble neurons. And also a lot of more recent exciting findings, a lot of them from the Susumu Tonigawa's lab, showing that these active ensemble neurons in some way are essential for expressing the encoded memory. So today what my goal is for today's talk, if you don't remember all the details of my talk, I want to there are 2 points I want to make. That is there's a heterogeneity within the memory engram. So we show it in 2 ways. One way is that the type of experience-dependent plasticity happening within the memory engram is not homogeneous. there are different type of experience-dependent plasticity happening to different group of neurons, different sub-ensemble of the neurons. And also, the role of those active ensemble neurons is not just a way to turn on and turn off the memory, sort of more like a very simplified the model proposed nowadays. Rather, these different components of the memory ensemble can fine-tune the output of the behavior based on what's written in the brain. So let me start to introduce the idea of the active neuron ensembles. Of course, when you talk about the ensembles, the first thing is how do you define them, right? How do you define the active neuron ensembles? So for this, let's look at what happens when neurons are activated. So what happens to the activated neurons is the postsynaptic receptors on the membrane are activated by the release of the neurotransmitter from the presynaptic terminals. And this will cause calcium influx, membrane depolarization and calcium influx. And this is a very critical step. With this cellular events, the receptors at the synaptic terminal can get modified. So there's local changes happened at those synapses to change the strength of the synapses. And again, these changes can be transient and can be very local. However, if you want to change those changes to be long-lasting, the signal have to be transmitted from the synapses all the way to the nucleus to turn on gene expression. As you can see that these cellular events allow us to define the neuronal ensembles in different ways. For example, traditionally, the spike of the neurons, the neuronal activity will allow people to record the ensemble activity using in vivo recording. So the active ensemble neurons are defined by their activity correlated with the behavior output. And more recently, calcium imaging use this calcium influx events to monitor which neurons are activated. And these methods are very powerful, very informative. In fact, it can tell you already from published work to tell you there are a lot of different diversity in terms of how neurons respond in terms of to the similar, to one particular sensory input. But the problem with this method is you can't really genetically tag those neurons, so that it's very limited in terms of how you can understand what's happening at the cellular level, at the molecular level. So we actually relied on a third method, which is take advantage of these gene expression events triggered by neural activity to develop transcription-based neuronal activity reporters to map those active ensemble neurons. So this activity-dependent gene transcription program is composed of this first group of genes called immediate early genes, IEGs. They are turned on very rapidly and within minutes, very robustly. You can see this with a very sharp curve. And if you look at this ensemble, there are some of the ones that a lot of people in this audience are familiar with, like FOS, ARC, and ZIF268. And also this, the majority of the genes on this first wave of IEGs are transcription factors. They can then turn on the transcription of second wave of genes that tend to function at the synapses and to turn on different downstream effects and modify synapses in very different ways. Because this group of IEGs, their activation is so rapid and robust, so their promoters have been used to develop activity reporters to label, to map the active neuronal ensembles. So now this is what we have accepted so far. For example, people use FOSS, some people use ARC. FOSS have been used most widely. So let's take a pause. So this type of transcription-based reporter system, so really it's very powerful, but also there's one issue with this is depending on what you use, you assume this is the ingram, this is the neuronal ensemble that's responsible for memory formation, and without thinking that there are all the other transcription programs that are also turned on, not necessarily within the same group of neurons, right? So what I'm trying to tell you today is for quite a number of years, we're trying to understand that each transcription, activity-dependent transcription pathways can potentially activate a distinct group of neurons, different, not necessarily the same ensembles of neurons, because each transcription pathway can turn on different downstream transcription targets, because transcription factors exert their downstream effect by turning on the downstream gene expression, so that different pathways and different neuronal ensembles can be modified differently by the same experience. And I will take one step further to show you today, hopefully you will be convinced, that because of this different type of experience-dependent modification, that different group of ensemble of neurons will play different roles in learning and memory. So quite a few years ago, we decided to think how we can really prove this hypothesis to develop a system. So we developed a activity reporter system we called a robust activity marking system called RAM. And this work was led by a very talented postdoc, Andres, who's now an assistant professor at University of Copenhagen. And Yoni, who back then was a postbac student, now he's a very productive ND PhD student at UCLA. And also a lot of other people in the lab also helped. So the RAM system, as an introduction, is the idea is the principle is very simple. So it consists of an activity-dependent promoter that drives the expression of reporter molecules only within the activated ensemble of neurons. And also the RAM system has a temporal control which will prevent the reporter molecules from expressing outside of your experimental window, but only within the experimental window that the reporter molecules will be allowed to express in the activated neurons. And the next slide is basically a schematic drawing of the RAM system. As you can see, that one thing I want to point out is the activity-dependent reporter of RAM is designed differently from other existing activity promoters. So instead of using the upstream promoters of IEGs, we decided to develop we designed the promoters that are bound and activated by those activity-dependent transcription factors. So there's a IEG transcription factor bound to the RAM promoter to drive the expression, in this case TTA. So for the temporal control, we use the DOX-OFF system. So the promoter drives the expression of the tetracycline transactivator, which binds to the TRE elements, tetracycline response element, and drive the expression of the reporters. And in the presence of DOX, this binding is prevented. So this is outside of the experimental window. We put the animals on DOX, and only when animals are off DOX and the recording, the labeling is allowed to happen. So if I have time at the end during Q&A, we can talk a bit more why we designed the promoter this way, but in short, the advantage of this is this will allow us to directly read out the transcriptional readout of each pathway rather than just monitoring the upstream activation of the transcription factor, which may or may not turn on the downstream gene expression. And another advantage of designing the system this way is that that allows us to design smaller, compact promoters that will be able to package the whole reporter system within a single AAV. So for today's purpose, we're going to focus on this one particular learning and memory task, and I know it doesn't require that much introduction. We use the contextual fear conditioning system that it's a hippocampus-dependent learning task in which you put the mouse in the novel chamber, and then it will explore to form the memory of this context. If during the same period of exploration you give them food shocks, then they will form a fear memory that's associated with the contextual memory. So it's two separate memories. And then later on, if you put the mouse back to the same cage, it won't explore rather than freeze because they remember this fearful environment. So the level of the time that the animal spends freezing, it's a readout. As the more they freeze, that means they remember this, the stronger of this contextual memory is because they recognize this is a not very safe environment. So I will give you an example of what we see in the hippocampus, what the active ensemble looks like labeled by the RAM system. This is, I'm using our prototype RAM promoter that it's already published. I'm going to talk about other promoters in the later of my talk. So we inject the RAM virus into the dentate gyrus in this case, using the reporter to drive the expression of MCAT, a red fluorescent protein, in this case nuclear localized. And this, you can see this is after fear conditioning, and compared to home cage, you see an increase of number of the RAM positive cells. And every cell in the dentate gyrus is infected with the reporter, as you can see, as seizure can induce most of the cells. And then you wonder whether this labeling is specific for the contextual memory. So we did a number of things. I will show you one of the results. So we quantified the number of labeling, number of the RAM positive cells after different conditions. As you can see, contextual CFC activates labels a large number of cells, a lot more than the home cage, usually around 4 or 5%. And the reason we know this labeling is specific for the contextual learning is because of these two controls that we put in. So in case that the animals, as I mentioned, the contextual fear conditioning, in this case, animal forms two memories. One is the contextual memory that's formed regardless of whether they receive food shock. And the other memory is the fear memory, and the fear memory is not formed in the hippocampus, but somewhere else, but the food shock plus the contextual exploration will allow the association of these two memories. So as you can see, if you have the animals explore the environment without food shock, they will form the contextual memory regardless. So you can see the RAM system labels the similar number of ensemble neurons. And if you give the animals just the food shock without allowing them to explore the chamber, then they won't form the contextual memory, but they will have a fear memory somewhere else. So in this case, RAM doesn't label any additional neurons apart more than the home cage when they just receive food shock. So suggesting that the labeling of RAM is consistent with the sense that the labeling of RAND-labeled neural ensembles in the hippocampus is associated with the contextual learning. So this is one example. And the next example is relatively recently published example. I want to show you that how we use system to really get to the question, what happens during memory formation? What type of synaptic changes happen at the ensemble neurons, whether the learning-induced plasticity on the ensemble neurons is important for memory formation. So this particular example I give you focus on this region called CA3 within the hippocampus. So within CA3, it receives three major excitatory inputs: the MOSI fiber inputs from dentate gyrus, and another input from entorhinal cortex through the peripheral path, and they also form a recurrent connection among themselves called the recurrent pathways. So these three different pathways have played different roles during learning and memory. So the MOSI input is shown to be important for memory encoding, but not retrieval. Retrieval depends on the peripheral path. And the recurrent pathway is important for short-term memory and pattern completion. And not only that, those three inputs are also spatially segregated, that the MOSI input synapse onto this region that's very close to the cell body. And then the other two, the peripheral path, recurrent path forms at the more distal region. And not only they are spatially segregated, they are also morphologically very different. They are very different type of synapses. And at the distal region, and the synaptic structures that are very familiar to most of the people here, they are the typical dendritic spine structures. But at the MOSI fiber synapses, they are one of a kind. They form this very unique structure called Thorny excrescences. They are the multi-headed spine structures that are very complex, and they are very gigantic. Basically, each MOSI fiber forms a gigantic synapse with one of those structures, and then with many different release sites. So one contact with many synaptic release sites. So it's a very complex spine. So as you can see, that the function of these three inputs are very well have been extensively studied, but so far it's not very clear which pathway it's, even though behaviorally people singled out that the MOSI fiber input is important for memory formation, but it's not unclear what type of synaptic plasticity happen at any of these synapses are important for memory formation. So we thought it's because people haven't been able to really zoom in onto the neurons within the CA3 that are involved in memory formation. So that we decided to put the RAND reporter within CA3 and label the active ensembles within CA3 after CFC. As you can see the quantification here, that actually the signal-to-noise ratio of the RAND reporter is really good within CA3, and with very low background at home cage and very robust labeling after CFC. So my postdoc, Eddie, decided to really ask a very obvious question, say, what type of synaptic changes happen on these active neurons in CA3. So he decided to just patch onto the positive neurons and measure their excitatory inhibitory synaptic inputs and compare to the unlabeled neighboring neurons, that those are the ones that were not activated during the CFC. And what he saw, what you're seeing here is the miniature spontaneous like mini iPSCs. I'm just showing you this. He didn't see any difference in the mini iPSC, the inhibitory input. All he saw was the very robust, very striking elevated mini iPSC frequency on the RAM-positive neurons. The quantification is shown here. See, it's almost doubled the frequency. And then when he selectively blocked input from MOSI fiber input, you can use a drug, in this case, this LY drug selectively blocked the MOSI fiber input, and the difference between the positive and negative cells is completely abolished. So that means that learning selectively, and this is the amplitude. So that suggests that learning selectively strengthens the MOSI fiber input. And this is the pathway has been implicated during memory formation. And also not just, you can see the difference at the electrophysiology level. You can also see them at the morphology level. So Rodrigo, my postdoc, decided to sparsely label those in the CA3 regions and then quantify the, those Thorny excrescence size on the positive and negative neurons, as you can see, positive and negative neurons. And then again, very consistent with, because these synapses are known to be sort of increase their size when their strength increases. As I told you, they have a lot of release sites, and as they get bigger, they add more release sites. The structure gets bigger. So you can see the size of the RAM-positive, the TE, the Thorny excrescences size on those positive neurons are significantly bigger than the negative neurons. So that means it's like learning selectively strengthens MOSI to CA3 synapses. So, so in the interest of time, I was going to go through this published work, but I think I wanted to get on sooner to my to the more exciting stuff. So how do we know this learning-induced plasticity is important? And so I'm just going to skip through a lot of data to show you that in the case of studying MPAS-4's function during learning and memory, we found that MPAS-4 selectively, the molecule that Ken just mentioned, another IEG, that selectively modulate the synaptic input from MOSI fiber to CA3. And we know that its function in CA3 is selectively required for memory encoding, and it's not required in CA1. And not only that, when you don't have MPAS-4, what happens is that at the basal level, the Thorny excrescences are already enlarged, so they are saturated. So when during learning, this learning-induced strengthening cannot happen. The neurons the synapses cannot be strengthened even further, so that contextual memory formation is impaired. As I show you here, again, we do the same experiment. We put the reporter into CA3. In this case, we also take out MPAS-4. As you can see on the left, it's the control. As you can see, the mini iPSC frequency is higher in the positive neurons compared to negative neurons. And again, it's blocked when you block the MOSI input. When MPAS-4 is deleted, as you can see here, now everything is even the negative neurons, the mini iPSC is totally elevated. So it's saturated so that you can't really tell which neuron has been activated during learning, which neuron is not activated during learning. So this just gives you an example that using this type of reporters that will allow you to really go down to the cellular, single cell, cellular molecular level to really understand what happens during learning at the synaptic and molecular level. So now I want to sort of, for the rest of the time, I want to sort of start telling you the exciting stuff. As I mentioned to you, we developed this system. We really wanted to really tackle the question of different neuronal ensembles because the different activity-dependent transcription pathways potentially can activate different group of different ensembles of the neuron within the memory engram. So one thing I want to point out is if you just use the false reporter or arc reporter, even MPAS4 reporter, you are looking at a portion of the ensemble, not the whole ensemble. So the question is whether there are different subgroups of ensemble, different group of neurons within the memory engram, and whether there's heterogeneity in terms of function within the memory engram. So for this, we developed a series of activity reporters to label neuronal ensembles defined by different transcriptional pathways. So, so my student Xiao Chen, together with Max and Mei Zhen, decided to start out to really look at whether there's any evidence, before we go do everything, whether there's any evidence that these different neuronal ensembles defined by different transcription pathways are different, right? So what they did is they put the RAN reporters, different RAN reporters into dentate gyrus, and then they label those ensembles using CFC. And then a day or two later, they use electrophysiology and behavior experiments to really see whether these different ensembles are different. And we focus on dentate gyrus, and dentate gyrus is known to be involved in pattern separation. And so we designed a very simple contextual contact discrimination assay. So we field conditioned animals in the context A, right? And then after that, they formed a contextual field memory in context A, and then we have them to recall, we ask them to recall the field memory in the same condition context A, or in a very similar but different context B. Basically, it's the same chamber, but with different floor. Or as a control, a very completely different neutral context C, which has different shape, different smell, different lighting, everything is different. So the animal can clearly tell these three contexts apart based on their freezing level. So they know A is a bad place, so they freeze a lot. And they know B is somehow related to A, so you can see they freeze at pretty high level, significantly high level. But they also know B somehow is maybe not quite the same, a little bit different from A. So they freeze significantly less in B than in A. And in the neutral context B, C, or even try different like C and D and very different ones, they always freeze at the basal level. So you can see, depending on how close the context is to the conditioned context, the animals freeze at different level, has this freezing level. So we hypothesize that within the memory ensemble, so there will be different components within the memory ensemble to tune the output of this field memory. So in a sense, this output of field memory requires discrimination and some generalization. So the balance between discrimination and generation is important. You can imagine if you don't have, if you have overgeneralized field memory, and that will lead to, so that's the basis for PTSD type of type of mental condition. So ideas that, so indeed what we found is different neuronal ensembles can really push the curve, push the curve to the right. So some of the ensembles will push the system towards discrimination, and some of the systems will push the system towards more generation. So this is what I'm going to show you. This is very exciting stuff that we've been working on for a few years now. So the two ensembles I'm going to share with you is, no surprise, one of the ensembles I'm going to share with you is the FOSS-dependent ensemble. And the other ensemble I'm going to share with you today is the MPAS-4 dependent ensemble. So remember I said there are FOSS-dependent ensemble and MPAS-4 dependent ensemble rather than FOSS ensemble versus MPAS-4 ensemble, because the reporters are designed as the transcriptional readout of FOSS and MPAS-4. So the promoters are bound by either FOSS or MPAS-4 to drive the expression of the reporter. So why do we focus on these two? So of course we have other reporters, but we want to start with FOSS because this is such a classic and well-known pathway, and it's also known that it's known to regulate neuronal excitability, and it's required for LTP. And MPAS-4 is the gene that we study, and we know a lot about MPAS-4. We know it regulates inhibitory synapses onto principal neurons. Not always true. The example I didn't show you in CA3, it's affecting excitation, not inhibition. But in dentate, we found it's affecting, it's recruiting inhibition. So it's, from the outside, we know these two ensembles could be very different. So I'll just show you how they look. And using, again, the contextual field conditioning, as you can see, we put the reporter into the dentate gyrus, and then we put it in, and then during this off-dox period, we put animals through CFC and label the neurons that are activated during CFC. As you can see, and this is, we call it FRAN, FOSS-dependent RAN, and NRAN, MPAS-4 dependent RAN. They both label a group of neurons after CFC compared to home cage, and it's DOX dependent. When you're in the presence of DOX, you don't get any labeling, and also they are activity dependent. As you can see, we with this type of from the technical detail, with this type of reporters, you always have to titrate, because the virus-based reporters, you have to titrate so we titrate the titer of the virus so that the ensemble size of these two is correlated with what we see by when you label FOSS and label MPAS-4 in dentate gyrus after CFC. So the ensemble size is between 1% and 2% of the total DG neurons. And we also, of course, I want to, also a very important question is whether they are independent ensembles, right? So does the FOSS dependent ensemble also depend on MPAS-4 and vice versa? So for this, we use the FOSS conditional knockout and MPAS-4 conditional knockout. We put the Cree into knockout FOSS or MPAS-4. As you can see, this is FRAM, and we take out FOSS in the, if you take out FOSS, the FRAM activity after seizure is almost diminished. In this particular case, there's some residual of labeling. So we think it's because there are other FOSS family members can also drive this reporter under very extreme conditions such as seizure. And when you take out MPAS-4, the FOSS FM activity is not affected. And when you take MPAS-4 out of dentate gyrus, you completely wipe out the NREM activity, and while the FREM activity is not affected at all. So that suggests that they are very independent. They are not dependent on each other. So another question we get quite a lot is a lot of people found it's very hard to believe because they always assume all the IEGs should be in the same population. In culture, a lot of times you can't get to those kind of specificity, and if you say what we did in the Greenberg lab, we dump KCl on the neurons, and all these IEGs are induced in all neurons. But in vivo, there are a lot of selectivity. So the question we got a lot is, are these two ensembles really a different group of neurons, right? And so this is a hard question because technically we cannot look at the both ensembles at the same time because they are both DOX dependent. So to address this question, we decided to use the FOS trap mouse. It's a FOS Cree-ER mouse where FOS drives the expression of Cree-ER, and then the Cree will turn on the reporters. So again, this is slightly different. This is looking at using the upstream promoter of FOS to label the ensemble. In this case, we use Chernolausen, and so supposedly the FOS ensemble, Cree-ER ensemble is in green. So we think maybe the FOS-dependent ensemble will have a lot of overlap with this FOS Cree-ER ensemble. If we label the FOS FREM ensemble in red, and you can see that they have a lot of overlap. And if the NREM, the mPAS4 dependent ensemble is a different population, then you will expect very little overlap with the FOS Cree-ER ensemble. And this is what we see, and quantification is shown here. So the NREM, you only see 5 to 6% like overlap with the FOS Cree-ER ensembles, while the FREM ensemble, not 100% overlap, over 50% of them are overlapped with the FOS Cree-ER ensemble. So this is another evidence suggesting they can be physically different. And then want to go a bit further, say, because the one hypothesis we start out with, these different pathways will induce different experience-dependent synaptic modification. So we decided to see whether they have different synaptic properties, these neurons. So what we did is we patch on, in this case, FOS, like FREM positive neurons, and record synaptic activity on these neurons, mini EPSCs and mini iPSCs, and compare to their neighboring neurons. And what we see is the FREM neurons, they have higher mini EPSC frequencies. No difference in the mini IPS, not difference in the inhibition, only the EPSC frequencies. The NREM neurons, the mPAS4 dependent neurons, on the other hand, doesn't change excitation at all, and it recruits more inhibition onto those NREM positive neurons. That's another evidence suggesting that they are different. Yeah.
So those changes are after learning, right?
Yes.
So it doesn't say what the difference between the neurons that will be FREM positive or NREM positive. It says what are the differences after learning?
Yes. So that's something that we are working on it to see whether they are different beforehand or whether they are intrinsically or extrinsically different, whether they are different group of neurons genetically to start with, or it's a stochastic event that assign them to different group of neurons. That's a very a question that we want to address. Yeah. So so this is another evidence, and then and the another thing we did was to really think if this group of neurons. So another example, this is one hypothesis that they are modified differently by experience, right? And then whether they have different roles in learning and memory. So to start getting at this question, we decided to first look at whether their activity the activities of these ensemble neurons are different during the period when the animals have to perform the discrimination. So what we did is we used the reporter to drive the expression of GCAM6F. So basically, you express the GCAM in those ensemble neurons, and then we measure the sort of population activity using photometry. And so what we did was, so you're going to see this graphs quite a lot. So we use a CFC to label those ensemble neurons to express GCAM in those neurons. And then we put them back on dogs, and then we put them back to A and B and C. And during the exploration memory retrieval, we monitor the activity of those ensemble neurons. So what we see is the FRAM, the force-dependent ensemble neurons, they show high activity. They are excited in, when they are recalled in A and B, and more than C. So they seem to be, the activity level is high in both A and B. So the question is, what's your guess for the NREM neurons between A and B and C? So it's very surprising to us. Now we are not surprised, but back then we were surprised. And what you see here, the NREM neurons, it's more excited. The activity is much higher in the context B, which is not the original condition context. It's a context very close to A, where they really have to turn on the discrimination. They need to be able to tell that B is not similar to A. So this is the quantification. As you can see, that the FRAM neurons are almost equally activated in both A and B. And the NREM neurons, on the other hand, even though their activity is higher in A than a neutral context, but what's surprising is the activity is much higher in a context B, where the discrimination is required. And this the activity of those NREM neurons in B is actually correlated with their discrimination activity. So what I plotted out here is the activity when they are in B, so the discrimination index quantified based on the activity level. So activity level in B minus A normalized by A plus B, and versus the behavior discrimination, which is the freezing level difference between A and B normalized by A plus B. So you can see this superactivation in B is correlated with their ability to discriminate between A and B. So this is very exciting. And the FRAM, the activity is not correlated with the discrimination. So based on this data, we hypothesize that the FRAM, the force-dependent ensembles, is probably involved in some kind of generalization, so without sort of discriminating between A and B, treating A and B the same. And the NREM neurons, the impulse-dependent ensembles, are important for discrimination. So to directly test this hypothesis, we decided to manipulate the activity of the NREM and FRAM neurons by using DREAD. I'm sure everyone knows the channels the neurons expressing those channels, their activity can be modulated by the ligand called CNO. So I'm going to show you, we did both the excitatory and inhibitory version of DREAD expression in the ensemble neurons. I'm going to just show you the inhibitory version. So we again put the reporter into dentate gyrus to express DREAD. This is an example of showing the expression of the channel. And again, this is a very complicated scheme. Basically, we label. Just if you look at here, you label the ensemble during the CFC, and then after 2 days, you put them back on dogs so that, and then you have them recall the first day, first recall in B, and then after 6 hours, recall in A. And then 30 minutes before the recall, IP deliver the ligand, CNO or vehicle to those neurons to manipulate the activity of those neurons. And the second day, with the same animal, we perform the recall in the neutral context C and again the context A. So it's within animal comparison, and as we can see. So what we see is when we inhibit FRAN neurons, the animal's ability to discriminate A and B is improved. So suggesting that the activity of the FRAN neurons are promoting generalization. And only affects the two-way similar context, doesn't really affect the discrimination between C and A. And the NRAN neurons, when they are inhibited, abolishes the discrimination, as you can see. Now the animals treats A and B the same. So suggesting the activity of the NRAN neurons is required for discrimination. So not just the freezing level, we also plot out the discrimination index. As you can see, it's really quite robust, the effect. So now the question is, what we want to address is, why is the FRAN neurons, what's the mechanism behind this generalization and discrimination? So remember that FRAN neurons recruits more excitation. So now we next want to ask, where does this excitatory input come from? So in the dentate gyrus, the excitatory neurons, the dentate gyrus granule neurons receive three major excitatory inputs from lateral entorhinal cortex, medial MEC, and hilus. This is through the Mossy cells. So we perform the dual patch. You patch on both the positive and negative neurons and stimulate these different pathways like LPP, MPP, and MCF. And we can adjust design the stimulation protocol to be selective to target these different inputs. So as you can see that we know we are targeting those input because if from the PPR, you can see that they have their characteristic short-term plasticity. So LPP is known to be have a short-term facilitation. The MPP, the input from the medial frontal cortex, has the short-term suppression, and MCF is again it's a potentiation. And also the input from the MOSI5 from the MEC can be blocked by DCG4, another blocker. And as you can see, we are very surprised again. Not surprised, but it's very exciting to see that the excitatory input that's recruited onto the FRAN neurons, they are selectively coming from the medial entorhinal cortex here. And this is the plasticity, the synaptic change, actually it's really in ways of increase the number of synaptic contacts from the medial, from MEC to the dentate gyral cortex, based on there's no change in pair pulse ratio, there's no change in MPA and MDA ratio. So again, this is already saying this type of plasticity is slightly different from what people, the Hebbian type of plasticity that people are more familiar with. And then the question is this input from MEC important for memory generalization? So the input from MEC is always known to be spatial, give the input the spatial information into the dentate gyrus. So we decided to put DREAD into into MEC. And as a control, we also put DREAD, inhibitory DREAD, into LEC. As you can see from the fiber trap of those ensemble neurons expressing DREAD, you can see our targeting is pretty good. So the MEC forms the terminal onto the medial molecular layer, and the lateral LPP form the terminal onto the outer molecular layer of the dentate gyrus. And consistent with our hypothesis that the MEC input, excitatory input from MEC is important for generalization. So when we inhibit the MEC, you get this increase in discrimination. Similar, recapitulate with what we saw when we inhibit the FRAM positive neurons, right? And we know this is specific for MEC. When we inhibit LEC, it doesn't change the discrimination and generalization. So this is generalization. So how about discrimination on the mPAS4 dependent ensemble? So I told you mPAS4 ensemble selectively recruit inhibition. And we all know there are different types of inhibitory neurons. And in dentate gyrus, there's the major ones are PV neurons, CCK neurons, and SST interneurons. And so we wanted to know which interneuron input is being recruited onto the NRAM positive neurons. So my first hypothesis must be the PV cells. But again, so we decided to express chernodopsin in all the interneurons using the GET2-CRE lines. This is done in the GET2-CRE mouse. And then again, we use the dual patch, both the positive neurons, negative neurons, and then we use the chernodopsin to activate all the inhibitory neurons. As expected, the NRAM positive neurons has a much significant, like a very robust, like much bigger optically induced iPSC amplitude. So this is the quantification. So when we do this experiment, if we flush in the blocker to selectively block the PV neurons, agotoxin, as you can see, this difference is preserved. But if we do the recording in the presence of conotoxin, which selectively blocks the CCK expressing interneurons, this difference is completely blocked. And not just that, we also use another drug, Wnt drug, which is to activate because the CCK neurons express the CB1 receptors, so the endocannabinoid signal can block the release of the transmission of CCK neurons. So the Wnt drug also abolished this difference between the positive and negative neurons. So this is just PPR suggesting, again, we know that this difference is due to an increase in the number of synaptic contact between CCK neurons and the NRAM positive neurons. So now the next question is whether the CCK neurons are important for discrimination. So for this, we use the CCK-CRE lines, but also we know CCK also expressed in the dentate gyrus granule cells. So for this, we use a genetic intersection method using the CCK-CRE line, and then we also use a DLX56, which is a GABAergic neuron-specific promoter to drive the CRE-dependent inhibitory DREAD. And you can see, it's hard to see here. I can just, then we can sort of really narrow down we quantify about 80% of neurons that are targeted are CCK positive neurons. And when we block the CCK neurons, just like as we inhibit the NRAM neurons, the discrimination is abolished. Not just that, it's the effect even stronger. As you can see now, what you see here, even in the neutral context, in context C, now you start to see the animal to freeze above chance. So suggesting that not just, there's a balance between discrimination and generation, and not just blocking generation, you also promote block discrimination and also promote generation. And this is specific to CCK because the PV and SST neurons, when they are blocked, they don't affect the discrimination and generation. So just to summarize what I told you, that during the contextual memory formation, what happens in dentate gyrus is that different group of, there's a neuron memory engram. There are groups of activated neurons, and some are FOS dependent, some are mPAS4 dependent. And we know there are different populations and they have different synaptic properties. And the efferent neurons recruits more excitation from entorhinal cortex. And the NREN neurons selectively recruits inhibition. The efferent neurons not just selectively recruit input from the medial entorhinal cortex to promote generalization, and the NREN neurons selectively recruit inhibition from CCK to promote discrimination. So, so one working model for us wanted to see, again, how does this circuit mechanism really affect the behavior output. So now it's a little bit hypothesis driven. So you can imagine what happens is downstream in CA3, the efferent neurons might have direct access to the contextual memory stored in CA3, as I show you, that marked by the RAM positive neurons. And the NREN neurons, what they do is they drive the feed-forward inhibition toward onto the memory engram in CA3. This feed-forward inhibition is known to be important for discrimination. So our hypothesis is this group of neurons directly drive the feed-forward inhibition towards the memory engram in the next station. So again, so just to summarize, and I show you there's two examples that there are different ensembles within the memory engram. They have different functions, different properties. And again, we wanted to understand all these the ensembles defined by other transcriptional pathways. There are a lot of unanswered questions. And again, the most obvious question is being asked is what determines their activation? And how do you decide which cell is going to be different ensemble? And whether they're determined intrinsically or extrinsically? And whether the different ensemble, how they do they really encode different components of a memory? So this is what I told you, and I show you the RAND system, and I show you that I hope I convinced. It's only two ensembles I show you, but I hope I convince you there exist distinct neuronal ensembles, and they're modified differently. They have different functions to change the output of the memory, and with this system and this approach can really get to the bottom of the question, is what's the underlying cellular molecular mechanism for learning and memory. And I already run over time, and the people in my lab and my collaborators that are involved in the work that I show you, and the funding and my lab. And I